Well, what a day. It's been a great time getting to see everyone and see everyone's talks. My name is Eric Jones. I'm a postdoc at Simon Fraser University, and I've been working with David Sivak and experimental collaborator Will Ludington at the Carnegie Institution for Science. And we've been grappling with this question of how do organisms acquire their gut microbiomes. And humans are, of course, an organism, and there is great interest in understanding how humans acquire their microbiomes to begin with. How do we go from a child, a newborn, with a very meager microbiome to the fairly robust set of microbes that we have in our guts today? And the human microbiome is really as unique as a fingerprint. You could sample all of our guts and none of them would be even close to the same. And we've started to appreciate over the last few decades that all of our microbiomes are relevant for human health. So for example, some microbiomes obviously play a role in disease. So the obvious example is like a pathogen. If you ever get food sickness after going out to eat, or if you get C. diff infection, this is some pathogen is in your gut and it makes you ill. We've started to get a more holistic view of the microbiome as well. For example, we have a set of healthy commensal microbes that actually help us metabolize or help train our immune system. This isn't a single microbe, but it's rather a collection of microbes. And on the contrary, there are so-called dysbiotic or imbalanced microbiome compositions where you have too many of some microbes and too few of another's. And these dysbiotic microbiome conditions are associated with diseases like obesity, Crohn's disease, or even type 2 diabetes. And so there's great interest in understanding how we got the microbiomes that we have. Of course, answering this question in humans is hard because we have a lot of microbes and we're always being exposed to microbes, and trying to figure out how my gut changes from yesterday to today is a very demanding task. So instead, I'll be working with the fruit fly as a model ecosystem for microbiome complexity. And these fruit flies have, in nature, typically around 10 microbial species, and we'll be considering a set of microbes of only 5 bacterial species. And these will be lactobacillus species, which are responsible for making yogurt, and acetobacter species, which are responsible for making vinegar. What's nice about these fruit flies is they can be reared germ-free without any type of bacteria in their guts, and then we can dose their food with certain bacterial combinations, in this way associate them with any bacterial combination. And in the past, we've used the fruit fly and we've given them different microbiomes, and we've shown that their physiology drastically differs depending on their microbiome composition. For example, their lifespan can vary by as much as 10 days based on whether they have one microbiome composition or another. And so to understand this microbiome assembly process, what we do is we take a set of flies and we feed them all some combination of bacteria. Here, for example, we're feeding the flies some orange, purple, and blue bacterial species, and then we wait 10 days. Coming from a physics background, I of course refer to bacteria by colors rather than their names, but their names are here on the bottom legend. They're lactobacillus and acetobacter species. And the idea is that we do this for 48 fly replicates for this particular combination, and then we crush them up and we ask which microbes were in their gut. Most of the time, these bacterial species colonize with an abundance of between 10 to the 4 and 10 to the 6 viable bacteria per fly. But sometimes the bacteria fail to colonize entirely, even though we are really dosing these flies with a high abundance of bacteria. They still fail to colonize. And so to understand this stochastic assembly process, the fact that even though you feed them bacteria, the bacteria don't necessarily colonize, we consider the presence and absence of these types of bacteria. In this way, we can build up a multinomial distribution of colonization outcomes, where we can look at the probability that all fed species colonize, that 2 out of the 3 fed species colonize, that 1 of the 3 fed species colonizes, or that none of them colonize at all. And then the experimentalists in Will's lab repeated this experiment for all possible 31 combinations, a so-called combinatorial dissection. And all of these data points from the roughly 1,500 sacrificed flies are summarized in this figure here. The way to read this figure, in the x-axis is the combination of fed bacterial species, and the y-axis is the probability of colonization of each of these possible colonization outcomes. So, for example, If you look at this bacteria, this combination here with this yellow and purple, this says that when yellow and purple are fed, they colonize something like 80% of the time. The probability that only yellow colonizes is about 10% of the time. The probability that only purple colonizes is about 5% of the time. Each one of these columns contains a multinomial distribution, and therefore the dots in each column sum to 1. You can look at this plot for a long time. I know I have. And you can start to ask questions about it. For example, maybe I care about the experiments where 2 bacterial species were fed, and I want to know how yellow performs. And so we could look for 2 species fed. Yellow's colonization ability is something like 90%. This is interesting because when we lay out the data like this, we start to notice patterns. Some of these bacteria, like the purple, are very weak colonizers, colonizing 60 or 70% of the time. Other bacterial species, like orange and green, colonize basically perfectly. Furthermore, this yellow bacteria, for example, colonizes perfectly when fed on its own, but as soon as you give it some other bacterial species that you feed it with, it starts to fail to colonize. And so we notice that these colonization odds are both species and diversity dependent. Going about our everyday lives, when we ate our delicious lunch this afternoon and perhaps had some microbes on a fork, the probability that those particular microbes colonized are probably much lower than these colonization odds because we really blast these flies with bacteria. So we really think the stochastic microbiome assembly is happening into all of us all the time. Additionally, we find that these colonization odds are context specific. This means that the colonization odds of some bacterial species are facilitated or inhibited by the other bacterial species that you feed alongside. For example, this purple bacteria is facilitated in its colonization ability when it's fed alongside orange and yellow. On the contrary, this purple is inhibited by the presence of blue and green. Where on this watermelon plot, the green indicates facilitation and the red indicates inhibition. If we sort of squint our eyes, we can notice on the bottom right of this heat map, we have this block of magenta. And this corresponds to Acetobacter species, the purple, blue, and green, being inhibited by other Acetobacter species. This is probably the effect of something like competitive exclusion. All the Acetobacter species are competing for the same resource. Or perhaps over-acidification. These acetic acid bacteria metabolize more acid than is good for them. On the other hand, we have on this green block on the bottom left, these Acetobacter species are facilitated by the presence of lactobacillus species, these orange and these yellows. This is probably the effect of some type of metabolic cross-feeding where lactobacillus species can break down their yeast food into a nutrient that the Acetobacters can take advantage of. On the whole, we can look at these context-dependent interactions, which are calculated only using presence-absence data from these microbiome experiments to infer ecological interactions. This is especially cool because presence-absence data is pretty easy to get for anybody that does 16S sampling or even culturing of bacteria. So what's the point of all this? One natural implication of these results is thinking about medicine for the microbiome. This fact that a lot of there are many diseases associated with dysbiotic microbiome compositions. One type of therapy that's been used recently is to try to modify somebody's gut microbiome state in order to change their gut microbiome composition to a more favorable state. For example, by using probiotics or fecal microbiota transplants. What we argue is that stochastic microbiome assembly underpins probiotic and fecal microbiota transplants. For example, just because probiotics or fecal microbial transplants might not work all the time, perhaps some of their failure to cure a disease is because the wrong microbes are not sticking, or all of the microbes that you want are failing to stick. Based on this, we have some design principles for microbiome therapies. For example, if you're constructing synthetic therapies where you're able to put whichever microbes you want in that particular therapy, you should prioritize microbes that are strong colonizers, include microbes that facilitate each other's colonization, and include multiple microbes that can form the same important function to sort of build in functional redundancy. Additionally, if you know the colonization odds of different species, you can calibrate dosing regimes so that you know how many doses of a probiotic, for example, a person ought to take. The last part was trying to model this process, basically assuming that the colonization odds of each bacterial species is an independent process, And we can use this model to basically recapitulate the majority of the multinomial distributions that I showed a few plots ago. If you take into account context-specific interactions, you can do even better. And with that, I'd like to thank you for your time. It's been great to see all of you today, to watch your talks and see your posters. I'd like to thank Will and Jean, who are also part of this project. And if you'd like to learn more, I'll refer you to the paper out earlier this year, and you can contact me by email or Twitter, and I welcome any questions.